Today we are going to talk about a very, very exciting subject, which is structure and function of potassium transport system. So, why this subject is so exciting? The subject is so exciting because potassium plays such an important role, I would say important roles in plant life. So on this slide you see written different plant functions that potassium participates in. It includes elongation, growth, defense, osmotic regulation, maintenance of protein conformation, and so on and so forth. So why it is so important? Mainly because it stabilizes protein synthesis, and it is involved in activation on enzymes and of course maintenance of osmotic pressure. And it neutralizes negative charges on proteins, and it is important in pH homeostasis. And as we have already talked about, it is important in maintenance of membrane potential along with proton pump. So that's the main functions of potassium. It is actually a macronutrient, so it comprises up to 10% of plant dry weight. And it is involved in virtually everything that every aspect of plant's life. And sophisticated potassium transport systems are expressed in all tissues of plant. So here you see a diagram of Arabidopsis plant, and here you see the names of different transporters that are expressed in different tissues. So you see the transporters that are localized specifically to guard cells that are involved in closing and opening of stomata. Very important transporters are located at the root, and they are involved both in acquisition of potassium and in transport of potassium into xylem, and into storing potassium as well. So you know that potassium concentration inside the cell is much higher than outside. So it is about 100 to 200 millimolar, and inside, and about 10 to 200 micromolar inside. So concentration inside the cell is about 1,000 times higher than outside, but you know that membrane potential in the cell is negative, so the driving force, electrical driving force compensates chemical driving force, gradient difference. So as I already mentioned, potassium is very important for the life of the cell. So if external concentration of potassium is very low, then you need active transport to transport potassium inside the cell. And in high affinity range of concentration, potassium uptake takes place against electrochemical gradient. So on this graph you see potassium uptake in micromole per grams of fresh weight versus concentration. So this is rubidium, which is transported through the same channels as potassium. So they trace rubidium concentration in the plant depending on concentration outside. And here you see the uptake in a mutant that is lacking AKT1 channel, and this is uptake in the mutant that is lacking HAK5 transporter. So can you guess, can you figure out which of those transporters are high affinity, is high affinity, and which one is low affinity?
I think that ET-hex is high affinity because it doesn't bind with rubidium. Greater when the concentrations of rubidium are increased, and because rubidium is not the target ion for this channel.
So, what do you think? So you understand what I'm saying? These are mutants, right?
Mm-hmm.
They are lacking this particular transporters.
I think that AKT1 mutant is a high affinity mutant because in the interval of low concentrations we see practically no transport through it and no transport of rubidium.
And so it is high affinity, you think?
Yes.
What do you think? So, Alexandra.
I think AKT1 is low affinity and AHAK5 is high affinity.
Yeah, actually it's correct. So there are 71 potassium channels and transporters. They were identified quite a bit, and we will talk a little bit about the families of transporters. So AKT is a so-called shaker type potassium channel. And here it is. Of course, on this graph you see only small representations of potassium transporters. So, but AKT1 is important transporter that transports potassium inside the cell. And there's another AKT transporter that is transporting potassium into the phloem. Then there are cyclic nucleotide gated channel, which is by definition ligand, right? Gated channels. And here you see cyclic nucleotide gated channel that is transporting potassium into the root. Then work its outward rectifying channel and its high affinity transporters also in the root, shaker type potassium channel, and non-selective cation channels. So not all potassium channels are highly and exclusively selected for potassium. Some can let other cations through. Outward channel and TPK1 channel. This is not what I want. Okay, so potassium channels, majority of them are actually voltage dependent, for example, shaker, and they can be weakly voltage dependent and rectifying. This is inward rectifying, this is outward rectifying channel. And there are voltage independent potassium channel, so-called TPK1, which are mostly located on the inner membranes. So potassium transport is highly important, and so there are many different factors that control and regulate its activity. So including cytosolic calcium, cytosolic pH, polyamines, proteins, 14,3,3 proteins, and different calcium-dependent proteins, calcium-dependent kinases, calcineurin-dependent kinase, and calcium-independent kinase as well. So these are all cellular transporters in Arabidopsis. This is actually an example of Arabidopsis transporters. So here is the family of shaker type potassium channels, KCO potassium channels, HKT TRK transporter. In Arabidopsis there is only one. In salt resistant plants, salt tolerant plants, there are many more. HAC transporters and potassium proton transporter, antiporter homologs. So first we are going to talk about potassium shaker type channels. So first thing you should remember that it is first potassium selective and it is voltage dependent. They are involved in uptake of potassium in the soil, which is very important. Also, they are involved in potassium secretion into the xylem and transport of solute to the phloem. They are very important in stomatal movement, in cell elongation as well. That's how they look. This is a subunit which consists of 6 transmembrane domain and selective potassium pore. And the channel itself is 4 tetramers arranged around central pore that is selective for potassium. So that's another representation to figure showing the structure of subunit of the shaker channel. There is a voltage sensor, N terminus, C terminus, and a pore. And on the C terminus we have cyclic nucleotide binding domain, anchoring domain, and conserved hydrophobic domain. So all shaker channels have this structure, this type of structure. Depending on subfamily, the shaker channels can be outward rectifying and inward rectifying. So they are, as I said, voltage dependent, so they're activated either by membrane hyperpolarization or depolarization. So which ones would be activated by hyperpolarization? Hyperpolarization.
The outward channels will be activated by hyperpolarization.
Outward.
Yes.
Sasha.
I think inward.
Inward. If you have hyperpolarization, you have more negative side inside the cell. So as a result, they get in, and they're activated when they're hyperpolarized. And when cell is depolarized, the driving force would be, for potassium, maybe outside. Okay, clear?
Yes, it was like, I thought it's like in animals, because they have mainly outward channels for their potential in nervous system and muscles.
That's a very good point, but you should forget most of what you learned in animal physiology, because you remember that in plants, our driving force for membrane potential is very different from animal membranes, right?
Yes.
We have, plants have a proton pump, and that's why the membrane is always negative on the inside. So potassium is actually practically in equilibrium because there is less outside, but since the potential is negative, the electrical driving force is inside. Okay. Now, I have a small task for you. So, imagine there is AKT1 hyperpolarization activated inward rectifying potassium channel. Please design an experiment to see if this channel is regulated by cyclic nucleotides and present results in case of AKT1 is activated by cyclic nucleotides, and you can do it in any model system. We talked about xenopacilicides. Relatively, if you can do it in your notebook now, okay? You can look at the mutant, you can do whatever you want. But you want to draw IV curve. Понятно?
IV curve is...
IV. Напряжение ток.
Like in usual.
Yeah, the usual IV curve. Anyone ready? Inward rectifying potassium channel, right? Inward rectifying. Помните? Inward, hyperpolarization, IV curve. Okay, как бы вы стали, how would you approach this?
Probably if this is inward and outside we have the minus potential.
Inside.
Okay.
Hyperpolarized. This is, so the cell is always negative here.
So if it goes in, the potential should be changed from minus in the cell to plus.
And then it will go where?
And it will go up for a while it is working. So probably it should be kind of going like that in steady state, like in equilibrium. And after hyperpolarization, it should go up and
Out or in?
if we
Inward.
inward. And if we measure outside, we have the opposite situation.
Yes. So, okay, I'll just... So, we have 100 millimolar here, yes, and 100 micromolar here of potassium. Yeah?
Mm-hmm.
So, according to Nernst potential, how would you make potassium not move anywhere? You don't want it to move. Here it is 1,000 times less, it's opposite. Micromolar, millimolar. Yeah? So the outside is 1,000 times less potassium than inside. What do you do with, so what is the force for potassium if there is no potential?
Proton force.
For potassium. Here is 100 millimolar, okay? And 100 micromolar outside.
So there is no any involvement of proton force.
Right now, no. There is no membrane potential whatsoever on the membrane, and the concentration of potassium inside is 1,000 times higher than outside.
It won't go inside.
It is 1,000 times less outside.
So it won't go inside through inward channels.
It is 1,000 times less concentration outside than inside. Where potassium wants to go?
Out.
Out, not in.
But it is inward channel, so it's
No, we are not talking about channel at the moment. We just want to figure out what's going on, okay? So you have a strong driving force outside. So what do you do to make it stay outside? not go outside or inside. Just stay as it is. How you maintain such a strong concentration difference?
Using importers or active phases.
To do what?
To maintain the difference.
In what?
Membrane potential.
Membrane potential. Okay, so what membrane potential has to be inside so that there would be no driving force for potassium anywhere? It will just stay like this, 1,000 times more potassium ions inside than outside. How do you do it? How cell can do it? How cell can maintain such a huge concentration difference?
We can also use negative ions like chloride to make the kind of neutral electric force.
No, we can't. If it is neutral electric force, by concentration they will flood inside the cell, right? By the outside, because here's outside the cell, because there is 1,000 times high concentration, all right, inside.
Mm-hmm.
It wants to go outside, but something holds it there, inside the cell. What it can be?
It can be a positive membrane potential.
Negative.
Yeah.
Correct, Sasha?
Yes.
It is negative membrane potential inside. So it is minus, and that's why it's not leaving, because it loves its minus, okay? Plus loves minus, that's just the way life goes. So because there is a negative potential inside, the potassium doesn't leave. And that's, and the potential is so-called potential, equilibrium potential, which we talked about before. Unfortunately, we were sick. It is, you can use this equation to calculate the potential that will make potassium stay inside. All right, so for potassium it's about minus 60 millivolts. So as a result, negatively charged, when membrane potential is negative, all right, then potassium doesn't have certain negative potential. Potassium does not have a driving force anyway, because it stays in the cell due to the negative charge on the membrane. All right? Понятно так примерно?
Yes.
Cool. And this negative charge in plants is created by proton ATPs and other ATPs, but mainly by proton ATPs. So now we go back to our AKT1 channel. So it is inward rectifying. So in which square the current would be? Sasha?
Right.
Yes, please.
This.
Absolutely.
This is the resting potential. So this will be current. It's inward.
Yes.
And since this channel is hyperpolarization activated, there will be
Nothing here. Excellent. And how would you show that it is regulated or not regulated by cyclic nucleotides?
Well, we can use the patch cloud tests with or without cyclic nucleotides in the solution, and see if this curve varies in the experiments.
So would this point change with your addition of cyclic nucleotides?
No.
Sasha, would this resting potential change if you add activating or deactivating agent for the potassium channel?
I think no.
And you?
I think also no.
Excellent. It wouldn't, because it does not depend on how active channel is, right? It depends exclusively on the difference in concentrations of this ion across the membrane. Good. So can you draw the line if cyclic nucleotides activating?
Activating this, channel, it will go like this, I guess.
Yeah.
And if it's repressor channel, it will go like this, or like this.
Yeah. Is it clear?
Not really.
Okay. Can you please explain it? So here is that's our current, right? The inward current. So this the steeper it is, the stronger, the higher current is at certain potential.
Okay, like it's kind of something of sorts similar to fermentative kinetics.
No.
I mean, my association, like when it has inhibitor or activator, it has similar changes when we have line curve.
It's not quite, because what is the difference, Sash, between, say, transporters and channels?
Well, transporters have the limit of activity,
Like enzymes.
Go like this. This is the amount of
Substrate.
substrate, and this is the activity of enzyme and channel, and mode A, mode S, and channels go like this because the amount of active channels only regulated. It goes like this or like this, and enzymes they go like this. Yeah, they go like this because it has the limits. And transporters do so also. Not much of ions can pass through one transporter because they are working really slowly. And when the activity of an enzyme is limited by an amount of active enzyme, so there is a limitation, there is a plateau on this curve of enzymatic or transporter activity, and channels does not have that. All of the ions can pass through this similar, relatively little amount of channels. So even channels, quantity of them is only affecting the slope of this line.
So
if there is enzymes or transporters, the quantity will affect the height of the view. So it will be like this or like this.
is it clear? The
Yes.
channels are pretty much holes, okay? They just either open or closed. So it is very different. Thank you very much. Thank you very much. Okay. So, shaker channels. So, for shaker channels, there are certain groups of shaker channels. Some of them are outward rectifying, and you immediately, by this graph, you can tell outward or inward, right? No problem. And what about this one?
It's kind in both of inward and outward channel.
So it is voltage regulated, but less than these other groups. You see, so this AKT2 is regulated, but not as much, and it's more like inward rectifying. And you see, they show it with membrane, negative membrane potential. Yeah. So it is a normal cell with the membrane potential, because you see that resting potential is like in a negative range. So in the group 1, it is our favorite AKT1 and SPIC and AKT and others, and there is a group 2, and both of those groups are inward rectifying. And okay, I'm talking just about Arabidopsis, because for Arabidopsis we have a really good information, and if I would talk about all the plants it would be too confusing, basically. and there are outward rectifying channels as well. So these are groups. So how we distinguish those groups? One of the ways to distinguish is the presence of anchoring domain, which mediates protein-protein interactions. It's one of the most common motif in structural motif in many proteins. And here what it looks like. And some of the groups have anchoring domains, some of them don't. So like these two groups can be distinguished by the presence or absence of anchoring domain. And in the silent AKT KC1, there is no anchoring domain. And finally, there is anchoring domain in GORC. And it's very important to understand the structure of the channels and separate them to different groups, because when you look at the plants from other groups, other plants that we probably don't know much about, we can immediately deduce how many different shaker channels do they have, how many belong to different groups, and we can guess their function in the plant. And it may be very important because, for example, we can't find really good salt resistance in Arabidopsis, whereas in Gallophyte plants we can find it, and maybe they have some specific potassium channels that can help us to design salt-sensitive crops. But how would we know where to look at in the genome of those plants for the channels that we need? Okay, that's how we do it. We look at the genome of this Gallophyte plant and find, say, potassium or sodium channel, and then you have several candidates you can examine. Also, so what they're doing, the shaker channels, AKT2 is involved in potassium loading and unloading in the phloem, and it controls, it is involved in controlling membrane potential, so in phloem. This is AKT2. So the group 2, which is AKT1 and other heteromers of AKT1, they are major pathway for the potassium inside the cell, inside the root cell. The group 2, KAT2, it also is involved in loading and unloading into the phloem. And GORCs are expressed in root hairs, and they are playing a role in osmoregulation. And the score, which is this, it contributes to the xylem, secretion into the xylem. So you see they all have their own specific roles, and according to the roles is their expression and regulation. There is this SPIC, here it is, group 1 of shaker channels, and it's a major pathway for uptake of potassium into growing pollen. And you understand that it is very important for the uptake of potassium is very important for elongation of, say, pollen or of elongating root cell, because when you uptake potassium, it increases osmotic potential, it drives water into the elongating cell. So this is very important. And KAT1 and KAT2, which are group 2, and they main conductance of, they are also very important in potassium conductances. All right, so they are regulated, and their regulation is different for different channels. And for example, acidification reduces potassium currents, and Some of them are not just permeable for potassium. For example, spic and from Arabidopsis and rice AKT1, they have a measurable conductance to NH4. Again, external acidification reduced potassium current through group 5 channels, and also voltage gating strongly sensitive to external potassium. So in general, they have a very high variability in the voltage that activates them, from minus 50 to minus 100. Calcium is very important in regulation of potassium channel activity, and they can block, internal calcium can block potassium channels at high concentration. Whereas some channels like SCORE, which is this outward rectifying, proposed to be substantially permeable to calcium at low external concentration. So we have talked in the paper about this, right? And this is just to remind you that how calcium can regulate potassium channels. So this is regulation of AKT2. You remember this mildly voltage regulated channel, and when calcium increases, activates calcineurin B-like protein, which activates calcineurin interacting protein kinase, which phosphorylates the protein. But in this particular case, this complex increases AKT2 transport activity by facilitating its introduction into the plasma membrane. And the same group, I mean, there are different calcineurins and different calcineurin interacting protein kinases, but in this case we are talking about AKT1. And AKT1 activity is significantly enhanced at low potassium by this complex. Interestingly, calcineurin on its own can activate AKT1, affect the activity of AKT1. This kinase, when activated by CBL1, it specifically interacts and phosphorylates AKT1 channel. So here is the latest paper on the structural basis of regulation of this AKT1. It's quite interesting. There are two serine residues, one on serine 338 and another is in 26. So here is one and here is another, and they're being phosphorylated. And this is a line of different channels, AKT1, 2, 6, KT1, different potassium channels. And this is these is those serine residues. So they're specific for AKT1, you see? There are not homologs in other channels. And one is an N-terminal and another in C-linker. And here is the results. What can you say, Sasha? Looking at the results, this is expression in Xenopus, all sites.
You can see that when single AKT1 is expressed, then we can see no response, and when it is expressed with CPK the CBA, we can see that we can see iron covenants.
What about, what about this? What it tells us?
It tells us that change of phosphorylation site in this channel changes its activity and it decreases.
Pretty much is abolished at 23, yes, 26, and 33, 38, and it's very much diminished in 26.
Yes.
Correct?